Okay, so I'm going to talk about the molecular genetics and molecular physiology, if I may coin that word, of nodulation control in legumes. And as I've indicated or hinted before, the work is done within the Center for Integrative Legume Research. And before I get cut off at the end by the chairman, I would like to acknowledge some of the people. Now, you're not supposed to read all of this, and I'm not trying to brag about all the people who are involved in this, but what is important is that I want to, please help. Okay, where's the pointer? On top, right? Yeah. I want to really highlight the fact that some people contributed over the years consistently. And I really want to acknowledge one of the people who is in the audience, and that's Brett Ferguson, Dr. Brett Ferguson, who has been the leading academic researcher heading up that group supervising, co-supervising people. And I especially want to speak about the work of some of the people like Dougal Reed and Yu Chan Lin and Satomi Hayashi. But this is not minimizing some of the other names that you can see here. And some of the names go back maybe 10 years already and so on. Some of them are still present here now, like Dong-Soo Lee and Lena. So thank you very much to all of you. Many of you are in the audience. It's really, really important that we have worked together. I also want to go to the bottom of this large list and really thank the ARC for the Center of Excellence funding, because ultimately over a period of 10 years we got something like $38 million, and that was just wonderful to do a lot of the work that we were able to do. But also the Queensland Smart State Initiative, UQ Strategic Funds, Herman Slade Foundations, and so on were involved in some of the funding. Okay, the overall biological problem, and that's why I'm showing this slide here, which has nothing to do with nodulation control, is that organ formation, numbers of kidneys, numbers of eyes, numbers of ribs and all of these things, all of these aspects of organ formation is genetically controlled. Right? There are genes which make the decision of how many eyes we have. Now, it's not the genes which are making the eye, in other words, the morphogenesis of the eye, but there's a decision-making process using the morphogenesis of how many of these primordia will actually develop. And so therefore there is usually a negative regulation. The developing eye, for example, in the human, in the mammalian embryo, is controlling how many other eyes are being developed. And this, of course, sometimes goes wrong. As you know, people have extra fingers, as you can see here. This is a Chinese person, and it's only a few years ago, 4 years ago. And you can see that both hands have extra digits. And as a matter of fact, what is interesting is that whatever goes wrong in the one hand also goes wrong with the other hand. So this is a commonly controlling mechanism, but it also happens on the feet. So therefore, this person has something like 30 digits altogether. Quite normal otherwise, but just simply numerous things. So these aspects are, of course, fantastically biologically interesting. I mean, how does this happen? Why does a plant, for example, make a single leaf, or sometimes makes triple leaves, and sometimes it makes 15 leaves, right, in a pinnate type of plant? Why do we have flowers which have 4 petals, and sometimes with 8, and sometimes with 12, and so on? So this comes up again and again, and of course it becomes economically important, because you have to ask the question, how many flowers, for example, does a tree or a plant produce? What is controlling that organ formation? What is controlling these aspects? So therefore, we are fusing biological interest, economic interest, because many of these things are significant, and focusing maybe unnecessarily on us humans. It's also medically significant, because a lot of malfunctions in humans are caused by extra organ formation. Okay, now plants, and I have had people come to me saying plants don't really have organs. People have organs like livers and kidneys. Plants just have tissues, right? Well, plants, I think, have organs too. So therefore, a flower, a root, and a root tip, and so on. And the organ formation that I'm interested in is significant because it occurs on a large group of plants which are called legumes. And this organ formation is the formation of a nodule, of a root nodule. And these root nodules are de novo organs. In other words, the root as it develops will just simply continue to develop as a root. It will never make a nodule unless there is an inducing agent there. And the inducing agent is a bacterium in the soil, broadly called Rhizobium. Now, there are different Rhizobium species which are specific for different legumes, but that does not really matter. What matters is that the Rhizobium knows how to produce a signal which is structurally analyzed. It's a lipo-oligosaccharide signal. And this nodule, these cell divisions which make the nodule, and here you can see the vascular tissue of the root, and so the cross-section of the nodule, you can see it's highly organized. This nodule forms de novo in response to the Rhizobium. Now, the Rhizobium itself, which normally is a soil bacterium out here, has the advantage in inducing this organ because it knows how to invade the tissue. And this puffy area in here in the middle is actually the symbiotic tissue where the process of nitrogen fixation occurs. And nitrogen fixation, symbiotic nitrogen fixation, is a process by which atmospheric nitrogen gas, 79% of what we breathe in right now, in and out, we don't know how to use it. But yet that nitrogen is very, very important for every amino acid in our body, for every nucleotide, and for thousands of metabolites. So therefore, it's an essential element, but the only way that it goes into in the biological system is through this process of nitrogen fixation. And so the bacteria housed in this organ have the capability of converting nitrogen gas, N2, to ammonia. So by splitting the triple bond one by one by one, making to ammonia. Now, this process allows legumes, as you know, to grow on very, very nitrogen-poor soil. It grows on the beach. I mean, I've seen it. You walk along the beach, they're beautiful legumes. And normally, they're usually very, very green. They don't have nitrogen deficiencies. And so, therefore, legumes become very agronomically important plants. We all know about the importance of soybean and clovers and medics and chickpea and peas and beans and so on. And even trees, such as wattle trees and the legume biofuel tree Pongamia, are important for our society. Okay, so therefore these organs are very, very neat, and a lot is known, and I will touch on what is known and how they develop. But ultimately, the question is, how is their development regulated? Because what we know, as I will show you, is that not all the root system becomes nodulated. There is a selectivity of tissue specificity. So therefore, how is this regulated? Well, we know that there are external clues. And there's a whole set of lectures that I could give on that, and we had PhD students completing that. External clues such as nitrate, potassium nitrate, any nitrate, will inhibit nodulation. And this is sort of the plant being smart. Nitrate, as you know, gets converted to ammonia, and so therefore you have a nitrogen source. So therefore the plant says to itself, why make a nodule and have this whole process letting the Rhizobium into the nodule when I just can take up the nitrate and convert it to ammonia. So therefore it's an energy-saving step. So nitrate inhibits nodulation, and it inhibits nitrogen fixation. pH inhibits nodulation, right? Acid soils are not very, very good to have legumes on, right? I mean, they get inhibited in terms of nodulation. I'm not going to speak directly about this. I'm going to hint a little bit at the nitrate inhibition because of its involvement in internal regulation. And that's what I'm especially interested in. What is the internal, the native regulation by the legume? What genes does a legume have to actually regulate how many nodules it will form at any one time? And so therefore we are speaking about plant genetic control of the process. Okay, now the basic observation is very, very common. I'm sure that everybody in this room knows about this, that when you pull a legume out of the soil, as you can see here, you usually find most of the nodules on the top. It's usually on the taproot or on lateral roots which have formed along the top. And if you look at the lower part of the root system, there are almost none. And this is not controlled by the fact that the rhizobium cannot get down there or there's too little oxygen or so. As a matter of fact, if you're in a glass house or in a pot, if you don't inoculate this legume plant, and you wait until it has a root system which is all the way down there, and then you inoculate it with the rhizobium, it will make nodules on the bottom. So therefore, these roots on the bottom are totally capable of making nodules, right? But they don't under normal situation. And there's a reason for that. It is that these nodules up here, their physiological formation, anatomical formation, actually leads to regulation of further nodulation down there. So therefore, there must be some kind of communication and some signals. And I will speak to you about those signals and what we know. So what is our approach to finding this? Well, I grew up in a laboratory of William Hayes, Bill Hayes, who, as you know, was very, very important in bacterial genetics. And so therefore, I know about the importance of yeast and E. coli and even in Drosophila, what does genetics do for this? And those of you who are working with Arabidopsis, for example, in plants, also now know about the genetics of impact. So therefore, my approach in the early 1980s even so, was to use induced mutagenesis. And we were one of the first laboratory who said, let's do it this way. Let's make mutants of plants which don't nodulate or which nodulate differently, rather than what has been done before by the outstanding English scientist called Thornton. And then what he did is he isolated ecotypes which did or did not. Well, it's very, very nice to look at biodiversity, but the problem is that ecotype 1 versus ecotype 2 probably differs by 10,000 genes or something like that. And so therefore, you don't know what is the causative mechanism. So if you want to have the mechanistic approach, it is of advantage to actually disable genes. So therefore, we utilized induced mutagenesis, and this is work done back in Canberra when I was at the ANU, and I had a brilliant honor student and who then became a graduate student and then became even a postdoc. And that man is Bernie Carroll, who now, as you know, is member of the faculty here at UQ. So Bernie and I go back to do these stages there. So therefore, we used induced mutagenesis to real genetic components of organogenesis and get some unexpected results. And I just want to tantalize you and get your interest. This is a normal soybean nodulation, and you can see the soybean nodules here very nicely forming along the top, maybe 50, 100 nodules or something like that. Well, by changing one nucleotide in one of the genes of the soybean plant, you get a soybean plant which looks like this. So therefore, it's nodule next to nodule. As a matter of fact, this root and this root here, several roots, look like corn cobs. 69% of the fresh weight of the root is actually nodule tissue. So this is abnormally high nodulation. And I maybe should highlight some of the numbers rather than you're speaking about a lot of nodules. I believe that it's of value to have numbers, so rather than saying plus, plus, plus versus minus or something like that, we grew some plants, and Dong-Soo Lee, who is working in our laboratory for the last 10, 12 years, she volunteered to count the nodules. And so it was 25,000 or so nodules on a mature plant. So this is extremely high in terms of normal agricultural situation. Okay, so how does all of this work? And I will run through actually the work, but before doing this, just quickly for maybe a minute or so, highlight how the nodule actually forms anatomically. Well, it starts out at this phase, and magnification is all altered in here. But ultimately, the plant, the root, which is the blue material here, secretes flavonoids. And these are different flavonoids. Clover is different than alfalfa, is different than soybean, is different than pea. But these flavonoids, such as diacetin or genistein, are recognized by the bacteria, the Rhizobia, and they induce the formation of the nod factor, the nodulation factor. And as I said, this has been beautifully analyzed by Jean Denarier in France, and the structure has been elucidated. And this nodulation factor is perceived by the plant very, very specifically, and it is perceived in a region close to the root tip, yes, behind the root tip, not in the mature root. And so therefore, after adhesion and so on, invasion occurs via a process of root hair curling. Now, that's not all the time this way. Peanuts, for example, don't do it by root hair curling. It just simply goes through an epidermal crack. And so therefore, they're slightly altered avenues of invasion, but it doesn't matter right now. What is important to recognize are the overall processes, because now if we look at the root a little bit larger, and you have the vascular tissue here, the cortex, and the epidermis, that root hair being invaded elicits the bacteria in the root hair, elicits cell division, and these cell division occur in the cortex. Now, they may happen at different levels of the cortex. For example, in soybean, it's just below the epidermis, right? Whereas in clover and medicago, it is very, very close to the endodermis. So bottom line is that cell divisions occur, and these cell divisions get invaded, these new cell divisions get invaded by the proliferating infection thread. And the infection thread is driven by bacteria dividing, actually. They're still directly outside. I mean, it's just like if I swallow a marble and then a marble is in my stomach, that marble actually still is outside, although it's inside of me, right? But it has not penetrated me. Well, the plant actually controls this by engulfing the bacteria by a plasma membrane-derived membrane, which is called the symbiosome membrane eventually, and together with cell divisions which get initiated from the pericycle. So there are two foci actually happening, the cortical and the pericycle one. You have the outgrowth of the nodule, and as you can see by the black little spots in here, these are the bacteria. Now, I drew this diagram extremely distorted. There are 25,000 bacteria in a single cell of soybean. Right? So therefore, a soybean nodule has millions of bacteria in it, which produce the ammonia, which gets assimilated by the plant system and then converted into glutamine and other nitrogenous compounds. Okay, so therefore, the process is therefore as indicated here, and the question is, how does the rest of the plant know that it occurs? Well, there are some extra little steps which are understood in this. For example, the nod factor, as it is produced, we know it gets recognized by a lyse M receptor kinase complex, a dimer, called NFR1 and NFR5. This is very, very similar to disease recognition. A lot of plant diseases are actually recognized through such receptors. And this receptor, and I'm not going to go into detail because I have lots to talk about, communicates with several other genes which produce proteins which are involved in iron transport. So therefore, there are iron fluctuations happening in the epidermal cell, in the root hair cell. And these iron fluctuations lead to calcium spiking, which is recognized by another receptor kinase, CCAMK, which interacts with other proteins and so on. I will not go into detail. But the bottom line is that this results in the formation of extra cytokinin in the epidermal tissue, which gets secreted and goes to the cytokinin receptor, a specific cytokinin receptor in the cortical and pericycle cells, so that leads to cell division. So you have a process of extra gene expression called nodulin expression, both in epidermal and cortical and pericycle tissue, and you have the induction of cell divisions. And so this is the process by which the nodule gets formed. Now, a whole bunch of hormones which we're familiar with all have effects, either stimulating or inhibiting, and I'm not going to go into this, but there are a lot of frontiers. There's a nice little review article which was published by Brett and several of us, and it's worthwhile to read that, although it is now 4 years old. It pretty well has 80–90% of the information. So therefore, the question is, I mean, here you see the process. By which processes, by which mechanism are these processes of nodule formation? How do cortical non-stem cells and pericycle stem cells, how are they induced to form this nitrogen-fixing nodule? Well, as I said, we used mutagenesis, and we isolated 2 types of mutants. The first type of mutants were non-nodulating mutants. And those were relatively easy to isolate. And then we ask what are actually the characteristics of these mutants, and most importantly, what are the genes. And this is some work which was then carried out by Dr. Arif Indersunema, who was a PhD student in the lab and is now a postdoc. And he used positional cloning in order to isolate the genes which are necessary for non-nodulation, or the other way around, which are necessary for nodulation. And it turns out that these are the 2 receptors which I told you are at the root hair surface and which receive the nodulation factor. So we cloned the soybean NFR1 and NFR5 receptors. And this goes along with parallel work in Medicago and also in P and also in Lotus japonicus. So those are the down mutations, you know, where you destroy something because, and because you destroyed it, it no longer nodulates. Those are the kind of mutations which led also to the recognition of all of the other steps, including the cytokinin receptor and the calcium perception mechanism. We also isolated mutants which had up nodulation. And I showed you earlier a picture of it. The major type is the mutation which goes from this phenotype of crown nodulation to excessive nodulation like this. And I call this super-nodulation or hyper-nodulation. And it turns out that such plants which are super-nodulating, hyper-nodulating are also resistant to external nitrate. So nitrate is not as effective in inhibiting nodulation in such mutant plants. And this was originally published at a long time ago in PNAS and in Plant Physiology, and some of the people in the audience probably weren't even born then at the time when we published this. So I want to summarize what we know about the mutants which supernodulate. Well, the first thing is that it turns out that in our hands in soybean, all of the mutations were in the same gene. Right? Now, in part, this is because soybean is ancestrally duplicated. And so, therefore, other mutations probably occurred as well in other genes, but we couldn't see the phenotype because there was a duplicated genome. However, for the gene which we call GM-NARC, nodule autoregulation receptor kinase, we find that we have multiple alleles, and this number of alleles is much higher now, but ultimately all of them are leading to hypernodulation or supernodulation. This gene is a common type of gene because it encodes a leucine-rich receptor kinase, LRR receptor kinase. And this LRR domain is postulated to be recognizing a signal. Now, such LRR receptor kinases are involved in multiple biological programs, and there's one which is called CLABATA1, which is very, very famous because of its importance in Arabidopsis shoot development. And indeed, GM-NARC is relatively closely related to CLABATA1, not only in amino acid sequence, but also in general structure. You have an intracellular kinase domain, a domain which phosphorylates residues. You have a transmembrane domain, and you have a long leucine-rich repeat domain and a single transduction. There's a single intron in all leucine-rich receptor kinases belonging to this class, so that when we align the structures, You can see the clavata one from Arabidopsis and the soybean NARC here and the related clavata one from soybean. You can see that the single intron is in the same position. Now it has different length and different composition, but it's always a single intron. And the amino acid sequence of the coding sequence is very, very similar. Now, when we look at expression of the GM-NARC gene, we find that, interestingly, it's expressed in a variety of tissues of the plant. And you can see here the root tip at 16 days and at 28 days, or the unifoliate leaf, or the first trifoliate leaf and the second trifoliate leaf. You can see there's quite a lot of expression. Now, the shoot tip is very, very weak in expression, and that we can confirm all of this molecular determination by real-time PCR. We can confirm by gene fusion, where we take the promoter of the NARC gene and we fuse it to a Gus intron gene, and you can see here that it is expressed in the vascular tissue of the leaf. Now, this is a very, very interesting observation. We're speaking about nodulation control, right? But yet the gene is expressed in the vascular tissue. But to the rescue of it, it's also expressed in the root, as you can see here, very, very weakly of the phloem parenchyma regions, and this becomes relevant later. Now, when we utilize such mutants, and we know that they're single gene mutations, and we do an old classical type of biological experiment, which is reciprocal grafting. Right? So therefore, you take the shoot of the plant and you graft it onto a root, and the root on a shoot, and so on. We actually find something very interesting. So therefore, this is summarized here, and I always want to explain it because it's critical for the final model. If you graft a wild type onto a wild type plant, so you reconstitute a wild type plant, you get normal nodulation. And so therefore, there's two, which means 20 or so. If you do a super-nodulating plant onto a super-nodulator, you get many nodules, as you have seen in the picture. The interesting experiment is if you take a root from a super-nodulating plant and graft a wild-type shoot on it, you get wild-type nodulation. And the reciprocal, if you take a wild-type root and you graft the super-nodulating shoot onto it, then you get super-nodulation. So therefore, the take-home message is that the nodulation phenotype of the root is controlled by the genotype of the shoot. Right? So therefore, this means that there's long-distance regulation of nodule number in the plant. So how does this occur? Well, this is just to show you the alignment of the gene. And I'm showing this for only one reason. You don't have to look at it in detail. Well, maybe two reasons. One is that the kinase domain of peas, soybean, lotus, medicago, and so on, all is highly conserved. And this is the LRR domain. And also you can see high level of conservation. What it means, and what we have confirmed in multiple ways, and I will touch on it just quickly, is that what I'm talking about is true for all legumes. Even legumes which have not been analyzed have this kind of nodulation control, and wherever one has looked, one finds the super-nodulation gene, the NARC gene, and the systems related to it. Okay, that's the wrong one. Okay, so therefore, we have come up with a model, and this model is now almost 30 years old and so on. But what it says is that the root, after being induced by the nodulation factor, will normally proceed towards cell division and infection, which will come together, as I've indicated to you quickly, to make the nitrogen-fixing nodule. But these cell divisions, so this arrow is slightly bent, it is these cell divisions, these early stages, which will induce the formation of a signal which I labeled Q. I tried to be funny, and this is a Q, a signal which is going upwards. And so this Q is perceived by NARC and the nodule autoregulation receptor kinase, which, as I showed you, if you have mutations in that, if you knock out this activity, you no longer have this upper circuit. And so therefore, SDI, again, I tried to be funny. This was the time of Ronald Reagan and so on. And so I figured that Star Wars initiative could also be the shoot-derived inhibitor. So the shoot-derived inhibitor gets activated because of phosphorylation of some components, and this inhibits further cell division. So therefore, you have a regulatory process which works for, doesn't work for a few days, so therefore you get nodulation on the ground tissue. And after 3 or 4 days, when this circuit has established itself and the concentration of shoot-derived inhibitor is high enough, you will have further suppression of cell divisions. And this is overall called autoregulation of nodulation, or AON. So today we know more about this. Now the clues, the critical clues for what is the mechanism came through work of Dougal Reed, and there's a picture of him who completed a PhD and even won the Dean's Medal, I think, or the Excellence Award. And this was work which was published in 1991 and 2011 and 2012. And what we discovered was what is the nature of Q. Rather than being a cryptic substance, we found out that it was actually a peptide, a Klee peptide, very, very similar to what is seen in Arabidopsis for clavata-1, where clavata-1 is controlled by a peptide called clavata-3. So therefore, the model system which happens in Arabidopsis, and remember, Arabidopsis does not nodulate or do any of these things, right? That kind of molecular mechanism also is occurring inside the legumes. So what did we find out? Well, through work of Satomi and others, we found out that the expression of these peptide genes, which we call RIC1, RIC2, and NIC1, and RIC1 stands for Rhizobium-induced Klee peptide, and NIC1 is nitrate-induced Klee peptide. It turns out that these peptides are expressed in the root, just like NARC is. I showed you that earlier. But remember, NARC is only expressed in the leaf, and these guys are not. And so therefore we know that. When we look at the soybean genome, and soybean is completely sequenced, so therefore we can analyze these situations, we find out that the sequences for RIC1 and RIC2 and NIC1 are actually duplicated. And they are duplicated as indicated here by the gray arrows. Gray arrow means they're not transcribed. They're not active. It is only the red ones which are active. So therefore, evolution, the plant itself, has evolved in order to make sure that there is a precise number of genes which have a precise number of activities. What is interesting is that all of these duplicated copies are, of course, highly related, and you can see the numbers between RIC2 and its silent copy and so on is very, very high, and otherwise RIC1 and RIC2 are also highly actually related. Now, when we speak about this, these peptides, we have to define actually where does the peptide come from. And the peptide, as you know, is a small protein, ultimately. And in our case, it's a protein which is 12 amino acids long. Now, it is highlighted here in terms of its conserved and sequenced. And you can see that there are some amino acids which are indicated at this level, which are literally 100% conserved. This is a logo plot. And you can see that there are some amino acids within the peptide, this being the peptide, which are 100% conserved. Other ones are more variable. Now, the peptide itself is located in a protein called a prepopeptide. Right? So therefore, there's a protein which is about 100 amino acids long, and it is this 12 amino acid peptide region which is close to the C-terminal. There's also a highly variable region between Klee peptides, and there seems to be no rhyme or reason. But what is important is that there seem to be some amino acids, such as indicated here in the front and also in the terminal region, which are highly conserved. So therefore, we have to ask something about their function. But what is important is that you have this conserved region. Just to show you how this looks in real life, these are the amino acid sequences of several clean peptides from Lotus japonicus, soybean, medicago, and so on. You can see these regions that I've spoken to you about, this here being the variable region, and you come to the conserved region, and you can see how highly conserved the individual residues are in that area. Well, we're going to focus on the soybean clean peptides. And the first one which I want to speak about is NIC1. This is the peptide which is involved in the regulation by nitrate. So it's a nitrate-induced clean peptide. And when you look at a diagram of the nodulation, nodule formation in relationship to nitrate concentration, we know that as you come to 4, 8 or so millimolar nitrate that you supply to the plant, you get severe inhibition. And so you can see this curve here, the blue curve, shows you the response curve of soybean nodulation to nitrate. When we look at gene expression of the NIC peptide, of the NIC1 peptide, we can see that the curve literally goes like this. It saturates very, very early, and some work of Da Fang, who's in the audience, has shown that even at a lower concentration, lower than 2 millimolar, you have susceptibility. So even 0.6 millimolar already is responding in terms of the activation of the NIC peptide. Now, it is also a very, very quick induction. So when we look at 0, 8, and 24 hours, you can see that it jumps up very, very quickly after nitrate addition. However, note, which is noteworthy, that there's always a low-level constitutive expression. Now, this is in contrast to the RIC peptides, and it's maybe not the optimal graph, but the NIC peptides come up something like by about 24 hours, they're also induced. Now, RIC1 is induced early, as indicated here, and RIC2 at a later stage. Now, RIC2 continues to stay at a high level of expression when RIC1 is coming down, and we don't know as yet the significance of this differential regulation. So here are the classes of single molecules. Now, what makes these single molecules verified single molecules? I mean, why do we say that these peptides, because they're differentially expressed and they respond to Rhizobium and so on, why do we say that they have anything to do with the price of fish? Well, it is because of these set of experiments. When you take a wild-type root, a wild-type plant, and this is cultivar Williams, and you look at the number of nodules per individual root, not root system, but individual root, and this is because they are transgenic roots, you find that it has this kind of level. Now, if you take a super-nodulator, as here, NOD4 mutants, you can see that there's a highly elevated nodule number. Okay, so that's the wild type versus the mutant, the background information. If you take RIC1 or RIC2 and you overexpress it in that root, so therefore you produce an abundance of transcript, easily 100 to 1,000 times more than normally recognized, you completely eliminate nodulation. If you do that same overexpression in the super-nodulation background, you get no suppression of nodulation. And so this argues very strongly, and which now has been confirmed by Professor Kawaguchi in Japan, that the Cli peptide actually binds to the NARC LRR domain. Sadly, we didn't do it. They scooped us. But the Japanese researchers are very, very good at doing that kind of research. But we're working closely together. So therefore, this illustrates that NARC is downstream from the Cli peptide. And I wanted to show you some pictures here of some of the work that people are doing in the lab right now. And this up here is a Gus fusion, the reporter gene, which makes blue color, as you know, with the RIC2 promoter. And you can see that if you have an empty vector control, there is really no color on the root. But then if you have the proper construct and you look, you can see the induction in the nodule meristem and lasting later into nodule development. I've removed a lot of the pictures in order to just simply highlight that. So the conclusion is it's absolutely nodule meristem localized, and it's inoculation dependent. Without inoculation, it doesn't happen. In contrast, if you look at the NIC1 gas fusion, and that is over here, you can see that it's nitrate activated. So for example, this is an empty vector control. This is a no nitrate control. So you see that there's no induction anywhere in the tissues. But when you add nitrate, you do get induction. What is interesting is that there's a very broad spectrum of tissue expression, but there's also developmental control. And you can see this by this very, very beautiful picture here of the root system of the plant. And this is a mature region, and this is a root tip region. And you can see that the lateral roots, as they come out, as you can see here, the lateral roots express it, but not the main trunk of the root. And you can see that young growing areas are. So therefore, these seem to be areas where nitrate is being taken up into the plant, into the root system. Okay, now in collaboration with, in collaboration with researchers in this building here, especially Professor Rob Capon and his associates, and Henry Dafang, who is the master student, we synthesized the peptides which are needed for nodulation control. And at the initial steps, there were actually two peaks, as you can see, two major peaks, but some purification was possible, and so this was purified into a single peak, and mass spec analysis was done, and the exact predicted molecular weight of the amino acids which are there has been obtained. Now, the negative aspect, or the sad aspect right now, is that this peptide, although it looks like the active component, is not active as yet, because it turns out that it has to be modified by the addition of 3 arabinose to one of the prolines. And so therefore, this synthesis is progressing right now in collaboration with researchers at Sydney University. Okay, so therefore, what do we know is clear expression, suppression of nodulation is NARC dependent. We know the same result in other plants, which amino acid is needed. I showed you the conserved moieties in some variable, and so therefore, what we did some site-directed mutagenesis, where we changed individual amino acids and then asked, are you still capable of suppression? So that's a suppression assay, right? So the results were more or less like this. As a summary of results. So here we have the conserved regions. This is a clear peptide region, and even the C-terminal extension. As you can see, there are the 2 prolines which are conserved, but they're not part of the peptide. And what we did is we mutagenized in the test tube individual codons, and so that the amino acids is changed. And some amino acids we changed to different amino acids to see, what is the functional component of it, and so on. And this was published in 2013 by Dougal and the group. Now the results are not 100% interpretable, but what there's some clear guidelines which come out of the paper. We looked at the amount of suppression. So this is negative statement of the data. So therefore, 100% suppression means that the peptide is not doing anything other than its normal function. So therefore, it's normal. So it's a low suppression, as indicated here, right, which are therefore very, very critical amino acids. And if we look at the logo diagram, we find that I indicated it by an asterisk, and you can see that it is, yes, these 2 conserved proline in the middle, and these positively charged amino acids. But it turns out that other moieties, like this leucine here, or this arginine, which are highly conserved, as you can see, every peptide that we've looked at has them. Actually, you mutate them into another moiety, and it has really very, very little effect. Likewise, the other moieties, like this duplicated proline here, has very little effect. So there are still questions which we have and have to answer. When we use the computer in order to make a model, predict a model, and remember this is only a predicted model, we find that the peptide itself folds itself into a boomerang like that. Now you know that in pi-mole you can rotate this, and it's really a very good harmony between the charge groups along the backbone. And you can see why, for example, at the pivot of the horseshoe, you have to have glycine. You have to have a small amino acid, because otherwise the peptide cannot bend. So the second recognition which we had is that proline number 7 is modified by a triple arabinose addition, and that's controlled by another gene which is called RDN1 or NOD3. And so some extra work to be done on all of this. Maybe I skip over this. I want to make one little point, because we have a paper coming on this, and this is that there are super-nodulating mutants in bean. And here is a wild type and a super-nodulating plant. You can see the growth inhibition that is caused by the super-nodulation. And you can see the root system similar to what I've shown you for soybean. Lots of nodules over the entire root system and only crown nodulation. If we look at the sequence of the super-nodulation gene, the NARC gene of bean as compared to soybean, we see that it's in a very, very similar genomic environment. And indeed, bean and soybean are highly related, maybe 20 million years apart. And so you can see some identical genes in the periphery, as you can see here. So this supports not only the sequence similarity, but also the genome environment. And so therefore, you can make a diagram as indicated here as compared to lotus, medicago, and bean. Okay, so therefore we found some mutations in the gene, and so therefore the phenotype that you can see here is caused by this mutation. We can do the same experiment again, but in this case we overexpress the soybean RIC peptide in the wild type and in the super-nodulating plant. And you can see the same result as you had in soybean, right? A heavy suppression in the wild type, but when you have an absence of the NARC gene, no effect. So therefore, arguing further that we have commonality of suppression mechanism. Okay, now I have a few more minutes, and I will use those to speak about some other work, because I told you about the up signal. So what is the down signal? And so what is SDI? And this is some extensive work which was done by Yu Chan Lin, and was published extensively about 2 or 2.5 years ago. Since then, we have received an ASC Discovery Grant, and Dong-Su Lee is involved in this, and we're working heavily in order to pick up the things which Yu Chan left behind. If you go to our greenhouse, you will see that it very much looks like this. So therefore, it looks like a hospital, right? And indeed, these plants, these soybean plants, are getting intravenous injections of liquids. Now, the intravenous injection occurs through petiole feeding. So therefore, what is done is that the petiole of the plant, as indicated down here, is cut, and attached is a Tygon tube, which is evacuated from with air, and so therefore you can feed liquids into this into the plant. This has an altered sensitivity as compared to spraying or watering or any of the normal plant physiological methods. And just for fun, we added some pigment, and you can see the color even moves to the flowers. You have blue flowers. So what we have done is we take leaves of treated or untreated plants, mutant or wild type. We do water extraction of those leaves, and we feed them by this petiole feeding into a mutant plant. Right? So therefore, the plant doesn't know how to make SDI, and you're feeding it what may be SDI. And so therefore, what we're looking for is a suppression of the phenotype of the hypernodulation phenotype to the wild type. So therefore, making a phenocopy. And this procedure works because we fed glucose and we fed mannitol, and you can see here this is a position of feeding, and you can see that the fed substance moves in both directions, here even illustrating that it goes into the root. So therefore, you can feed a plant with these metabolites, including possibly SDI. This works even in other plants. It works, as you can see here, in tomato and so on. Here, tomato and kale and so on. Okay, so there are many, many data, but the key data are highlighted by this red box. And this is looking at the level of suppression of nodulation after feeding the water extract. Now, if you feed the water extract from a water blank also, you can see that there's no suppression. If you feed the extract from a wild-type plant which has not been inoculated, so therefore a plant which has not been challenged, so therefore it did not induce clear peptides, therefore it did not induce the SDI process, you also see no suppression. But if you take a wild-type plant which is inoculated, and so therefore is challenged, so therefore it should have SDI, if you take that plant and you feed it, you get suppression. The same happens if you use mutant versus wild type, and you can see that the mutant doesn't have the SDI signal. Now, in collaboration with people here in this building and so on, we progressed in order to do a purification of this extract, which seems to have that activity. And the bottom line, the highlight, the key experiment is shown here. This is after excessive separation. You can see different fractions and feeding the wild-type extract in the blue column as compared to the mutant extract. Now, a mutant extract will not have any suppression, and so therefore the columns, the green columns, are all the same, and they're an internal control for the methodology. But it turns out that if you look at fraction number 16, you see that the wild type has severe suppression, whereas the mutant does not. And so this was separated by LC-ESI mass spec, and it was looked at in both the positive mode over here and the negative mode. And so, and what we found is that there are several candidate peaks, but there's one candidate peak, very small, as you can see, compared to the others, which is absent in the mutant. And so therefore, we progressed and looked further at detail in this, and we used some computer software like Calc and so on, and SciFinder, and looked at this peak. And the best predicted, but not confirmed, right? The best predicted structure of this is like this. So this is a very, very unusual compound, and as you can see, it has a very, very complicated name and so on, but it turns out to be a known compound in plants. And it is actually a breakdown product of a triterpenoid saponin. So therefore, what does this do, how does it do it, and so on, requires some further work, which we are engaged in right now. So in summary, what we know is that we have elucidated through biochemistry, through genetics, through molecular genetics, genome analysis, we have elucidated the mechanism of legume nodule regulation. So we know that Rhizobia induce primordia, and these primordia produce RIC peptides, which move up as Q to the shoot, to the phloem parenchyma, where the induction of SDI occurs, and SDI comes down and suppresses further nodulation. We also know that nitrate, ironically, works in a localized way. So nitrate inhibition works through the NARC, which is expressed in the root, and so therefore you have the induction of NIC1, which then presumably produce a similar compound like SDI, also inhibiting nodulation. So we have a situation here that two developmental regulatory pathways are controlled in different tissues, but using the same gene, right? And using the same type of single compound, but with some diversity. So therefore, RIC1 is different than NIC1. So therefore, it's a very, very novel regulatory mechanism, but obviously which develops into nodule autoregulation. So the conclusions, I think I've spoken about extensively, and it's pretty clear to you, and hopefully we have some time for discussions and questions. So I close by saying thank you, and I hope we can have discussion. Thank you very much.